going to examine some of the formed elements here. In particular, we're going to look at the red blood cells. We'll very briefly cover the leukocytes. Those are white blood cells, but for the most part, leukocytes will be discussed in more detail when we talk about the immune system. Please note that red blood cells are called erythrocytes. And in fact, erythrocytes is perhaps the better term because red blood cells are not, in fact, cells, at least not technically. I've mentioned this, but I want to emphasize this. You need to understand this. There is no nuclei in a red blood cell. There's no mitochondria. For that matter, we don't have any of the machinery necessary to synthesize new proteins. Without DNA, we can't do transcription, and if we don't have transcription, we're certainly not going to be doing translation. We are not making new proteins. Red blood cells are, in fact, made from living cells. Okay, they're made from cells that I will refer to for right now as stem cells, and we're going to look at that process in a little bit more detail. But at the very last step of these red blood cell productions, the nucleus is expelled. And at that point, this is really no longer a cell so much as it is a sac full of hemoglobin. I want you to take a look at the shape. It is biconcave. First off, let me note this. These are fairly small cells. To put things in perspective, we used to do a lab when we were on campus where we would take cheek cells, epithelial cells, squamous epithelial cells from scraping a toothpick on a cheek, and you spread those out on the microscope, and we would use some techniques to estimate the size of those cells. Cheek cells are about 40 to 80 microns big, so micrometers in diameter. These guys are 7.5, okay? Very small cells. That's important because they need to squeeze through capillaries, which are tiny as well. If we turn this, you'll see this center here where it's less dark in color. And the reason for that is this region here is fairly thin. Okay, it's thin, it's part of the biconcave disc. Flip this over on the side and hack it in half. Do a transverse section of it, and you can see that biconcave disc a little better, and the thinness between these membranes. Okay, and that's actually pretty important in terms of increasing surface area, so that molecules such as oxygen can diffuse into the cell more easily and bind to the hemoglobin. Incidentally, carbon dioxide also will diffuse into the cell, and we'll have a chance to talk a little bit more about both of those gases and what they do once they encounter hemoglobin within the cell. The primary function of hemoglobin, of course, is to carry oxygen, but that is its primary function. A secondary function might be to, in fact, carry carbon dioxide. To kind of give you some numbers to think about, I've mentioned these numbers before, but let's kind of think about them in terms of what this actually means. If we have approximately 5 million blood cells for every microliter of blood, that's what an MM3 means, 5 million per microliter. So just think how small that is, just as a reminder. So our liters, our base unit, 1/10 is a deciliter, 1/100th is a centiliter, 1/1000th is a milliliter, and 1/1,000,000th is a microliter. Okay, so we're dealing with a microliter and 5 million cells fitting into that, just again as a quick reminder, because you'll want to know this in terms of nursing, because normally we work with If you're in the lab, you're going to be working with milliliters. If you're in the nursing field, you're going to be working with CC, which stands for cubic centimeters, and that equals a milliliter. And so when you have cubic milliliters, that's equal to a micrometer. I'm sorry, a microliter. Okay, similar kind of idea there. Lifespan of the cell is 120 days. Please make note that on average we have 30 days in a month, and therefore 120 days is also exactly the same as approximately 4 months. And the reason I point that out is because I have had students get confused, because I tend to switch back and forth between these time frames. I will sometimes say red blood cells live for 4 months. Sometimes I'll say 120 days. And sometimes people don't have that light bulb come on and realize that I'm talking about the exact same thing. So I want to highlight that and make sure you understand so that you're aware of what I mean. Now here's a little mind-blowing piece of information. All right, so let's think this through, because just crunch some numbers. 280 million hemoglobin molecules in just one of these babies. Just one of these guys. 280 million. Whoa, that's a lot of hemoglobin. And then considering we have 500 million cells per microliter, and probably on average about 5 liters of blood. All right, I'm hoping that's going blow your mind as you realize how many molecules of hemoglobin we have. And of those hemoglobin molecules, one hemoglobin, again the abbreviation for hemoglobin is HB, one hemoglobin molecule can bind up to 4 oxygen molecules. So hey, that's a lot of oxygen that we have the ability to carry. Good thing, because we use it. At the center of the hemoglobin molecule is an iron group trapped in what we call this iron heme, this ring structure, which I'm going to show you on the next slide. And that iron is recycled. So when these blood cells get old and we recycle them, the iron will be reused. Now, taking a closer look at these hemoglobin molecules, you should be aware that we actually have 4 proteins here. We call this whole thing a protein, but we have 4 subunits, 4 peptide subunits. This first one, let me kind of highlight it or color it a little bit in blue for you, just so you can see it. Right, I'm not doing the most beautiful job, but there you go. Okay, and right across kitty corner is another one. And then we'll color this other one. I guess we'll do a darker blue. That sounds good to me. And you can see again, kitty corner is another one. Now the reason I chose 2 different colors is because these subunits are 2 different types, and they are arranged out kitty corner. So we would have, in most adults, we have alpha, beta, beta. Okay, that's the typical arrangement there. And inside each of these, we have these heme molecules, which I'm coloring in yellow. In the blue, they're tucked in there back in the back. You can't see them as well, but I'll still color them up in yellow. When we take a look at that, here's what we get, is this ring structure here. And notice my nitrogens. Nitrogens are really good at binding to things like, lo and behold, my iron, which is found in the center. Now, iron is an interesting molecule. It likes to grab up oxygen. We have iron oxide What is it? 2? I think so. And that can change. We can change the state of that iron back and forth based on the conditions that exist. Now, I will make a quick comment that while most adults have alpha and beta, a fetus actually has the gamma and delta. What is the delta symbol? I don't remember the delta symbol. I think this is delta symbol. Something like that. Anyway, gamma and delta. Now, to get you to think in terms of molecular interactions, which, you know, we're always going back, we're circulating back, we're integrating this information, we're putting it together in one big whole. These hemoglobin molecules have a higher affinity for oxygen than beta and alpha. And I want you to kind of put your thinking caps on and ask yourself, why would the human species evolve so that the fetus has this higher affinity higher affinity gamma and delta? Thinking about that for a second. And I hope, as you think about it, it popped into your head and you're like, wait a second, where does the fetus get its oxygen from? A fetus in utero, are their lungs working? Of course not. They're getting their oxygen from the mother's blood. And so if we are trying to exchange oxygen from this hemoglobin to this hemoglobin, yeah, the fetus better have higher affinity, or else it's not going to be as effective at taking that oxygen from the mother's blood. And so that's kind of a cool and interesting thing to think about. Every now and then an adult will actually have gamma and delta, but most of us don't. Red blood cells are critical for their ability to carry oxygen. And when things go wrong, then they can't carry oxygen as well, which can cause a condition called anemia. Now, most people think of anemia as looking something like this. If this is normal blood cell count, then this would obviously be a very lower than normal. And clearly, if we don't have enough red blood cells, we're not going to be able to carry oxygen. However, the correct definition of anemia is that it is a condition characterized by the reduction in oxygen carrying capacity of the blood. And I want to emphasize that you can, in fact, have a completely normal cell count, completely normal, and still be anemic. Let me doctor this image up a little bit. Maybe I'll use this one because that'll work. We'll even do that one. That'll work better. Okay. If these red blood cells can't produce enough hemoglobin, then instead of being a nice bright red, they tend to be pale pink. And what this signifies, if they're the wrong color, if they're not dark enough red, what this is actually telling somebody is if they draw their blood and you look at it under a microscope and this is what you see is these kind of pinkish cells. Okay, this tells you right there that you don't have enough hemoglobin packed in there. Let's go flip back here really quickly. So if you're supposed to have 280 million hemoglobin molecules per every red blood cell, what if instead you only have 100 million? Well, hemoglobin is what gives red blood cells its red color, and so if you don't have enough hemoglobin in them, those cells are going to look pink in color, and we call that, they're going to be hypochromic. Hypochromic. They don't have enough color in them. Hypochromic. And this reflects the fact that they have too little hemoglobin molecules in each cell. And without that hemoglobin molecule, they still have a reduced oxygen carrying capacity of blood. Maybe we have plenty of cells, but they don't have enough hemoglobin. You're still going to be anemic. Okay, and that happens. Right? That happens. There's a number of different types of anemia. And I can get into some of them. I talk a little bit about them. They range from simply iron deficiency. That's probably the most common. Iron deficiency anemia. Okay? And that would simply be due to lack of iron in your diet, or perhaps inability to absorb the iron. You could have perhaps enough iron in your diet, but you're not absorbing it well. In addition, vitamin B12 is actually pretty important in the absorption and transport of that iron, and its use for red blood cell synthesis. And so those are probably the two most common. Those individuals who are vegetarian or who are more on especially vegan diets really need to watch for this, because the most common source of vitamin B12 is actually animal protein, whether that's meat or eggs or cheese or whatnot. So somebody who is vegan will often have to supplement this, or just be very careful in their diet. One common substitute is brewer's yeast. One of my best friends in high school, it's been ages and ages, but I used to spend a lot of time at their house, and they were vegan, and they would drink brewer's yeast in their orange juice. And so I tried it once, and it was the nastiest stuff in the whole world, but rich in vitamin B. So anywho, iron deficiency, vitamin B deficiency is most common. There's also something called aplastic anemia. This one is common for people who have had chemotherapy. It's where the cells in the bone marrow that make the red blood cells have been damaged by radiation or chemotherapy, and are no longer able to produce red blood cells. And then another type of anemia is hemolytic. As the name suggests, this would cause the lysis of the red blood cells, and one of the most common types of hemolytic anemia is sickle cell anemia. Okay, and so those are some that I want you to know. Let me add one more: pernicious anemia. Pernicious anemia is actually related to the vitamin B12 deficiency, but there's a slight difference. Vitamin B12 deficiency, if you're not eating vitamin B12, if you're not consuming it, then you can't absorb it. Well, pernicious anemia, you might be getting plenty of vitamin B12 in your diet, tons of it in your diet. Maybe you're even taking supplements. You're taking it in a pill form, and you're just loading yourself up with vitamin B12. If it doesn't get absorbed, it's not going to help you. And so pernicious anemia actually comes from inability absorb B12. And this actually is a fairly common condition with individuals who have had a gastric bypass surgery, where they end up not being able to absorb that B12 very well. And the solution for pernicious anemia our monthly injections of vitamin B12. And so those are the different types of anemia that I want you comfortable with and familiar with, and kind of programming those into your brain and being aware of those.